Hello everyone. Welcome to the session of Sexual Reproduction in Flowering Plants Part 7. Disclaimer: the images, text and other material used in our presentations are for educational purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issue, please contact us at sathi@iitk.ac.in and we shall remove the content immediately. Please note that the parts of the content may have been generated using the generative AI, which is totally experimental. So, welcome to today's session, and today we are going to study about seeds and fruits. Okay, that is a very important part of this chapter, sexual reproduction in flowering plants. So, today's learning objective, let us see what are the learning objective. To understand the role of the structure of seeds and fruits. So, we will study two things: the role and the structure of seeds and fruits. To differentiate between the non-albuminous and albuminous seeds, perisperm, periderm, and what is true fruits and false fruits. All this we are going to discuss about it today. Index. So this we are studying which part of the NCERT? We are studying the 1.4 section of the NCERT, and that is post-fertilization structure and event. Means after fertilization, what are the different structures that are formed and what are the events that are taking place, right? So the first structure that we are going to study about the seed, right? And second is summary. So this is the topic subtopic 1.4.3 seed in your NCERT book. Okay, seed. In angiosperm, the seed is the final product of the sexual reproduction. So sometime it can be asked, what is the final product of the sexual reproduction? Final product of the sexual reproduction. So that is the seed. Seed is the final product of the sexual reproduction. And how the seed is formed? Which structure transformed to seed? We have discussed it yesterday also. Like after the fertilization, what transforms into fruit? So ovary transformed into fruit, right? Ovary is transformed into fruit, and the ovule transforms into seeds, right? So this seed is the final product of the sexual reproduction. Now it is often described as a fertilized ovule. It is described as a fertilized ovule. Fertilized ovule means the egg is present in the embryo sac. The egg is present in the embryo sac. And where this embryo sac is present finally? It is present inside the ovule. Right? So when the fertilization takes place inside the embryo sac, fertilization of egg takes place, the zygote is formed. Where the zygote is present? Inside the embryo sac. And where this fertilized embryo sac is present? Inside the ovule. So that is why sometime it is described as fertilized ovule. Is it clear? Fertilized ovule. Seeds are formed inside fruits. So where the seeds are present? Seeds are present inside the fruits only. So whenever you cut any fruit, you cut apple, you cut guava, you cut papaya, you cut mango, and what you see inside it? Seeds, right? So seeds are formed inside the fruits always. A seed typically consists of the seed coat. A seed is typically consists of seed coat. So it is having a very hard covering. Generally, you must have seen the gram. Gram, in Hindi we call it chana, right? So brown one. It is having the brown color seed coat. And when you split the seeds of this gram, what you see? The cotyledons. And in between the cotyledon, there is a small embryo axis. There is a, yes, gram is the dicot. It is a dicot seed. So that is the having the two cotyledon. It is having the two cotyledon. And there is a very small embryo axis in between the two cotyledons. Okay. So this is a different parts of the seed you see in the case of any dicot. And even in the monocot also, the monocot is also having the seed coat. It is having the cotyledon, but instead of two cotyledon, they are having only one cotyledon. And they are also having the embryo axis and embryo axis. Yesterday we have seen the structure of the embryo axis in the case of both the dicot as well as in the case of monocot. So the cotyledons, figure 1.15a of the embryo, are simple structure, generally thick and swollen due to the storage of the food reserve, as in the case of legumes. Hmm. So the cotyledon you see here, the cotyledons, they are having what? They are having the storage of food reserves, storage of food reserves. And here, this is the castor seed. This is the castor seed. So they are having the seed coat. You can see the endosperm. And inside here, this yellow part, this part is the cotyledon. This part is the cotyledon. Okay, this is the cotyledon. And the rest part, this outer side, this is the endosperm. The covering of it is the seed coat. Right? Yes. We will have a separate lecture for the PYQs. Okay. Now my voice is clear, I hope. Yes, we will have a separate lecture with PYQ section. Okay. Okay. So the cotyledons, the cotyledon in case of this dicot, so they are having this reserve food material. Now, in the case of this monocot seed, this is the one of the monocot seeds. Monocot seeds, they are having this scutellum. This is the scutellum. Scutellum is having what? Scutellum is actually the monocot cotyledon. Scutellum is the monocot cotyledon. And you see the endosperm. You see the endosperm? The food reserve, the endosperm is having a lot of food reserve. So this food reserve comes to this scutellum. And some sort of food material is stored in the scutellum. Right? So that is provided to the cotyledon. That is now they are having the food reserve in the cotyledon as well. A part of the endosperm, they are having the food material in the cotyledon also. Right? So the embryo are simple structure, generally thick and swollen due to the storage of this food reserve, as we see in the case of legumes. Right? So cotyledon, two cotyledon in the case of dicot and one cotyledon in the case of monocot. Now we will study the monocot seed structure first of all. So monocot seed structure, they are having seed coat. They are having seed coat. Protective outer covering of the seed. What is the function of the seed coat? It provides protection to the seeds from the adverse condition. Yes, for the monocot, we are having maize, we are having uh, wheat, rice, Monocot is actually, as the name, mono means one and cot comes from the cotyledon. So monocot means the seed which is having only one cotyledon, that is called as monocot. Similarly, dicot, the di means two and cot means cotyledon. So a seed that bears up two cotyledon, right? The seed that bears the two cotyledon is called as dicotyledon. The seed coat is the protective outer covering of the seed, and second part is the endosperm, that is the nutrient rich tissue surrounding the embryo, providing for the food germination. This endosperm is having we have talked about lot about the endosperm, how the endosperm is formed. The endosperm is formed from the result of the triple fusion, the triple nuclei, 3N ploidy. It divide and redivide and forms the multicellular structure, right? And that is called as endosperm. So this is the nutrient rich tissue surrounding the embryo and provide food for the germination. Not only for the germination but also for the developing embryo, because the endosperm formation takes place earlier than the embryo. Okay, endosperm form first and then the embryo forms later on. Why? Because the embryo needs some nourishment. So once the endosperm is formed, the nourishment is given to this developing embryo, and small amount of endosperm is still left there, and that is used for the process of germination. Now the embryo that contained the young plant with a single cotyledon seed leaf. Single cotyledon means we are talking about the monocot. That is why they are having the single cotyledon. And when the seed germinate, this cotyledon comes out in a form of a leaf, and that is called as seed leaf. When the monocot seed germinate, okay, the small leaf come out, because that is the conversion of that cotyledon into the leaf, and that is called as seed leaf, right? And the later on the new leaf comes out from the plumule part. So here you can see this is the structure of a maize seed. This is the structure of the maize seed. Now what are the things here? Let us see. You can see this is the radical part. This is the radical part, and radical part is covered by a sheath like structure that is called as coleorhiza. A sheath like structure that is called as coleorhiza. It is having this embryonal axis. When you see this, this is called as the embryonal axis. I'll just draw with a dotted line like this. This is the embryonal axis, whole this structure. And here you can see there is a plumule part. The plumule part is also covered with a sheath-like structure. It is covered with a sheath-like structure which is called as coleoptile. So plumule is covered with a sheath-like structure which is called as coleoptile. Radical is also covered with a sheath-like structure which is called as coleorhiza. This is also the same seed. This is the side view. You can see a seed coat, the cotyledons, the shoot apical meristem, hypocotyle root axis, endosperm is there, root tip. So this is both the structures are called as monocot. This is the structure of the monocot seed. So yesterday we have discussed about the hypocotyle and epicotyle. Can anyone tell me what is the hypocotyle or epicotyle? Because we have discussed this yesterday. Below the embryonal axis is hypocotyle. Okay. So The cotyledon, actually this is the comparison of the cotyledon, what is called as hypocotyle, hmm? That is the hypocotyle, the cylindrical portion below the level of cotyledon is the hypocotyle. Below the level of cotyledon is the hypocotyle, right? And above the level, that is called as epicotyle. Okay, level of above embryonal axis, above the level of cotyledon is called as the epicotyle, right? So we have discussed about this yesterday, epicotyle and hypocotyle. But this is the description of the structure of the monocot. They are having the seed coat, they are having the endosperm, and they are also having the embryo. They are also having the embryo. Next part, the other part are coleoptile. Here you can see coleoptile, the protective sheath covering emerging shoot in monocot seedlings. Okay, this is the sheath like covering that is covering the shoot structure, or you can say the plumule part. Coleorhiza, protective sheath covering that is emerging the root in the monocot seedling. That is, they are protecting the radical part. Okay. Now they are having a very special structure that is called as scutellum. Specialized cotyledon in the monocot that absorb the nutrient from the endosperm. This line is very, very important. Specialized structure. This is scutellum. This is called as the cotyledon in monocot. Okay, scutellum is the cotyledon of the monocot and it absorbs nutrient from the endosperm at the time of seed germination. So this scutellum is having the food reserve that comes from the endosperm only. So you must remember this line. In the case of monocot, the name of the cotyledon is got scutellum. There is no difference. There is no difference between the cotyledon and the scutellum, but this terminology is used for the monocot only. Okay, monocot, one cotyledon, and this can be asked sometime in the examination, like the food reserve in the scutellum is coming from which part? It is coming from the endosperm only. So you must remember this point. We have discussed about this structure. Now we will see the dicot seed. Dicotyledon. It is having the seed coat, protective outer covering of the seed. Like the monocot, the dicot seed also having a protective outer covering of the seed. Endosperm, nutrient reserve tissue in some dicot seed, but it is often consumed during the embryo development. It is consumed during the embryo development. This dicot seed is totally, it is having the endosperm, and all the endosperm has been consumed during the embryo development. Nothing is left. Embryo contain a young plant with the two cotyledons, and that is the seed leaves. Okay, so the cotyledon, the first seeds that you see at the time of the germination of a seed, that is actually the cotyledons. And that is why this cotyledon is also called as seed leaves. It is also called as seed leaves. Remember this. Hypocotyle, the part of the embryo, it is a part of the embryo between the radicle and the cotyledon. So that is called as hypocotyl. That is called as hypocotyl. So this is the part of the embryo only, right? Hypocotyl. So this is the structure of a seed. You can see the dicot seed structure. This is you can say the gram, not the gram, but it is a, you can say, bean seed, rajma bean seed. Okay, and these are the two cotyledon. At this particular point, they are having the embryo. This is the position of the embryo, and there is a small embryonal axis. So here you can see this is again one of a seed, and this you can see the seed coat. This is the seed coat, the outer covering of the seed, that is called as the seed coat. And this is structure, this, the outer side, this is structure. This is the endosperm. This is the endosperm. And here what you see, the cotyledon. Inside it, you can see this part is the cotyledon, right? This is the hypocotyl structure. Hypocotyl means here you can see the root tip, or you can say the radical part. This is the radical part. Okay. And the cotyledon is this. So between the radical and the cotyledon is the hypocotyl. Hypocotyl. And here you can see the shoot apical meristem is also here. Shoot apical meristem. Right? So this is the dicot seed structure. Dicot seed structure. The other part, like they are having the radical also. The radical, that is the embryonic root. That is also called as embryonic root, that develops into the primary root upon the germination. So first time the root emerges out of the seed, and that root comes out in the form of radical only. The radical comes out and forms the first root, and that is the tap root. That generally forms the tap root, primary root of tap root. Okay, this primary root is actually the tap root. So this is the main root. This is the main root, and later you can see the secondary root. They can give out the secondary root like this, right? So the primary root is actually the tap root, and that is formed from the radical only. That is formed from the radical only. Very, very important point, right? Cotyledon, the seed leaves that store and provide nutrient for the embryo during the germination. So, remember one thing for the dicot seeds. In the case of dicot, the endosperm is totally consumed. The endosperm is totally consumed for the development of the embryo. And the seed require 2 times energy. First thing, for the development of the embryo, and second time they need energy or the nutrients at the time of seed germination. But in the case of dicot, all the energy is consumed. All the energy which is stored in the endosperm is consumed for the development of the embryo. For the development of the embryo. So the question arises, from where they get the energy or the nutrients during the seed germination? From where they get? So the answer is this: cotyledons. Cotyledon is something different from the endosperm. Yes, cotyledon is something different from the endosperm. So don't get confused. Don't get confused with this. Okay. Okay. So the cotyledon, seed leaves that stored and provide nutrient for the embryo during the germination. So for the development of the embryo, endosperm is there. Energy required for the seed germination provided by the cotyledon, and this is for the dicot. And why so? Because for the development of the embryo, all the energy, all the nutrients from the endosperm is consumed, right? Epicotyle. Parts of the embryo above the point of attachment of the cotyledon which develops into the shoot system. So this is a small part in the embryo, okay, small part of the embryo, and that is present at the point of attachment of the cotyledon, okay, above the cotyledon, which develops into the shoot system, So this structure we have already discussed out. Types of seeds. Now this is also a very important point. Types of seeds. Mature seed may be non-albuminous or X-albuminous. Two types of seeds are there. Non-albuminous, and it is also called as X-albuminous. What is the meaning of this? Albumin. When they are not having any albumin is a type of nutrients, okay? The mature seed, when they are not having any albumin, then it is called as non-albuminous or X-albuminous, okay? Non-albuminous or X-albuminous. So the seeds are here divided into two types on the basis of presence or absence of albumin. It is a protein. Albumin is a type of protein, right? And in the case, if we talk about this human blood, then this albumin is the most abundant circulating protein. Albumin. In the case of plant also, they are having this particular protein in the seeds. So mature seeds may be non-albuminous or X-albuminous. Non-albuminous seeds have no residual endosperm, as it is completely consumed during the embryo development. Yes, the albumin protein is present in the endosperm, and as you know, some seeds totally consume their endosperm. So no albumin will be present there, and that seed will called as non-albuminous or X-albuminous, right? And the examples are here. You can see pea and groundnut. These are some of the examples that comes under the non-albuminous or X-albuminous. Okay, so no residual endosperm. Residual endosperm means leftover, some sort of like endosperm is left behind, then that is called as residual. So here no residual endosperm is left, as it is totally get consumed during the embryo development. Can you tell me whether it is a dicot or monocot? Which could be the seed, which type of seed that consumed all the endosperm during the embryo development? Dicot or monocot? So this you can see the examples also. Pea and groundnut, they are the example of dicot or monocot. It is the example of dicot. Pea seed you can split into the two cotyledon. Groundnut, that also you can split into the two pieces, two cotyledons, right? So those are all dicot. So dicot are generally non-albuminous or X-albuminous. And just now in the previous slide we have discussed that in the case of dicot seed, all the endosperms are used up. Albuminous seeds. Now comes the albuminous seeds. Retain a part of the endosperm as it is not completely used up during the embryo development. Hmm. They retain the part of the this endosperm that do not get completely consumed. Okay, integuments. Do they degenerate? No, they do not degenerate. They are present in a form of seed cover. They are present in the form of seed cover, right? Integuments only. They are the integument, seed cover. Albuminous seed retain a part of the endosperm as it is not completely used up during the embryo development. And the examples: wheat, maize, barley, and castor. These are some of the seeds that they do contain some sort of endosperm after the embryo development. So they are having some residual endosperm. This albuminous seeds, they are having the residual endosperm, and the examples are wheat, maize, barley, and castor. So do remember the examples of the albuminous seeds and non-albuminous seeds. Also remember what is left behind in the case of albuminous seeds. Okay. Occasionally, in some seed, such as black pepper and beet, remnants of the nucellus are also persistent. So, why this particular line is in a bold form, highlighted in a bold form? Because this line is very, very important for your examination point of view. There are the two examples: black pepper and beet. Two examples are here: black pepper and beet, right? They are having the remnants of nucellus and they persist occasionally, right? So remnants of nucellus are also persistent, and that is called as perisperm. That is called as perisperm. This residual persistent nucellus is the perisperm, right? So do remember what is perisperm. That is the remnants of the nucellus. Okay, because generally the nucellus get consumed up. Generally this nucellus is consumed up, but in some cases they are they are present after the seed formation, and that nucellus, persistent nucellus, is called as perisperm. Examples are black pepper and beet. Okay, black pepper and beet. So this residual persistent nucellus is the perisperm. Integuments of the, so you got the question here. One of the question was asked to me. Integuments of the ovules harden and as a tough protective seed coat. So this integuments later on after the formation of seed, they form a tough protective covering, which is called as the seed coat, right? Very important point. Integuments of the ovule are hardened and the tough protective seed coat. The micropyle remain as a small pore in the seed coat. You can see a small pore or a hole-like structure that is present on the seed coat. So I'm just drawing a very simple diagram of a gram seed like this, and you can see a small pore-like structure like this. Today you can take a gram seed and you can see that. Hmm? A small pore-like structure. What is this pore-like structure? This is the micropyle, the remains of the micropyle, and that is present in a small pore on the seed coat. What is the function of this micropyle, remains of the micropyle? We all know that the micropyle at the time of ovule formation, this provide an entry to the pollen tube structure. The pollen tube get enter inside this ovule for the pollen tube get enter inside it, and later on the fertilization process take place because of that only. But here, why this remains is still present? Because this small pore like structure is present. And very well, O2 exchange, yes, very good. O2 exchange takes place, this oxygen get enter inside this, and also water at the time of seed germination, right? So this micropyle is very, very important. This facilitate the entry of oxygen and water into the seed during the germination. During the germination process, this oxygen and water is required. As the seed mature, its water content is reduced. As the seed get mature, its water content get reduced. The seed become dry. The seeds become dry, and they are relatively dry, like 10 to 15% of the moisture by mass. Or only 10 to 15% of the water is left there. So it is totally dry. Because why they want to keep their seeds dry? Because in that dry form, it can be stored for the longer duration. Otherwise, it can desiccate, right? So when the seed get, yes, desiccate means the fungal infection. Yes, the bacteria and fungi can attack on the seeds and they can destroy seeds. But when they are dry, this fungi and bacteria, they could not harm that seed, right? So as the seed mature, its water content is reduced and seeds become relatively dry. 10 to 15% of the moisture get reduced by mass. The general metabolic activity of the embryo slows down, and when the seed get mature, all the metabolic activities cease down, or you can say slows down. Okay. The embryo may enter a state of inactivity, and that is called as dormancy. So the embryo goes for a long sleep. It is something like that. The embryo is sleeping, right? Because no metabolic activity is there. No water is there. So embryo may enter in the state of inactivity, and that is called as dormancy. So you can say the dry seeds are in the form of dormant condition. Or if and when the favorable conditions are available. What are the favorable conditions? Adequate amount of moisture, oxygen, and suitable temperature. When returns back to them, they will germinate. They will germinate. Okay? This we are talking about the seed, right? This we are talking about the seed, but this hibernation in the animals is a situation when the favorable condition is not available, right? The metabolic activities is not totally stopped there. The water content is not totally zero there, right? So the hibernation in animals is something quite different, right? Embryo versus seed. What is embryo and what is seed? Embryo is present inside the seed. Seed is having lot many parts. It is having seed covering. It is having cotyledons. It is having embryonal axis. In the embryonal axis, they are having the embryo, they are hypocotyle, epicotyle. All these parts are there. But what about the embryo? That embryo is just a baby plant, which is having the this plumule and radical part. Not totally. If it sees down, it is the lowest stage. It is at the lowest stage. Metabolic activity completely stopped in the dormant stage? No. Okay, it is at the lowest stage, because still 10 to 15% of water is still there, right? But the movement of the molecules, the the other metabolic activities, like they take the oxygen and all, that is all stopped there, right? But once they get this adequate amount of moisture, oxygen, suitable temperature, they will germinate. They will germinate. And ovules matures into seed. This we all know, that ovules is the only thing that got mature in the form of seed, and the ovary develops into the fruit. The fruit you eat is the ovary part of the flower. Is the presence of soil compulsory for the germination? Have you seen the hydroponics? With the help of hydroponics, now you can like grow your vegetables, right? So it is not very compulsory, but yes, this is one of the very important part soil in which the plants grow, because all the nutrients that they require are present in the soil only, right? So that is why the soil compulsory, but you can provide some substitute to those plants. You can provide substitute, like we are having the hydroponics. You can grow vegetables in the form of hydroponics. But what about the trees? Can you imagine that trees are growing without soil? Yes, it is an artificial technique to grow plants. Yes. So that is why soil is very important. They can grow, they can grow. In fact, when you make sprouts at your home, what you do? You soak the seeds overnight. You take any moong dal or any like gram seeds overnight, and then you tie it in a clean cloth and wet that cloth for one day. What you see the next day? The seeds of those dals or the chana gram that get germinated. That get germinated. And if you leave it, the plumule also come out. But after that, they require their establishment. Establishment where? In the soil, right? So soil is important. But for like herbaceous plant, you can grow in your hydroponics, but not for the trees. Okay, the ovules mature into seeds, ovary develops into the fruit, and the fruit which you all eat, we all eat, that we are eating the ovary of the flower. Okay, so that transform ovule into seeds and ovary into fruit proceeds simultaneously. So we have to study about the fruit also. The wall of the ovary develops into the wall of the fruit, and that is called the pericarp. We all know that like ovary is having a covering. Ovary is having a covering, and that covering is called as pericarp. That covering is called at pericarp, and this ovary is only changing to fruit. So the covering of the fruit is also called as pericarp. The fruit may be fleshy, as in the case of guava, orange, and mango, right? Or it may be dry, as in the case of groundnut and mustard. Hmm. So we are having some fleshy fruits and some dry fruits. So in the For fleshy foods, we are having guava, orange, and mango. Okay, and all these fruits, outer covering, guava outer covering is the pericarp. Orange outer covering that you peel off, okay, that you peel off, that is also the pericarp. Mango, you take out that peel, right? So that is the pericarp. Then what you eat in the mango, that fleshy part, that is the mesocarp. That is the mesocarp. And the hard, stony, inside the mesocarp, there is a seed covering. That is the hard, stony part. And that is called as the endocarp. That is called as the endocarp. So see, this fruit is having the three covering: pericarp, mesocarp, endocarp, right? Okay. So the groundnut are also having that same thing. The groundnut, they are also having the same thing. Uh, Pericarp, mesocarp, and endocarp. We are having the mustard also. So these are groundnut and the mustards are examples of the dry fruits. The fruits may be fleshy, as in the case of guava and mango. Groundnut and mustard are the dry. This we have already seen. Many fruits have evolved mechanism of the dispersal of seeds, right? The fruits are having a special mechanism so that they can be dispersed out, and their seeds can also be dispersed out, and later on these seeds can grow. Recall the classification of fruits and their dispersal mechanism that you have studied in your earlier classes. Now there is a question. Is there any relationship between the number of ovules in an ovary and the number of seeds present in a fruit? This is a question from your book. Is there any relationship between the number of ovules in an ovary and the number of seeds present in a fruit? Let us see the answer. Yes, there is a direct relationship. The number of seeds in a fruit typically corresponds to the number of ovules. Okay, number of ovules that were successfully fertilized in the ovary. So the number of seeds means number of ovules. Now we will talk about fruits. In most plants, by the time the fruits develops from the ovary, the other floral parts degenerate and fall off. This we have discussed about, right? At the time of this, fertilization takes place, right? Ovary changes to fruit, all the other parts of the flower wither off or the fall off. However, in few species such as apple and strawberry and the cashew, thalamus also contributes to the fruit formation. Thalamus, that is the base part at which all the floral parts are attached. Such fruits are called as false fruit. Okay, the fruit that is formed from, not from the ovary but other parts like thalamus, so they are called as false fruits. Most fruits, however, develops into only from the ovary and are called as true fruits. Yes, I am coming to that point. Most fruits, however, develops only from the ovary only, and the fruits that are formed from the ovary, they are called as true fruits. So see this figure very carefully. What this structure is, this structure which is I am drawing here, what this structure is. This is the ovary, and what is this? This part which you eat, that is the ovule, that is the thalamus, that is the thalamus, and these are the ovules that transforms into the seeds after fertilization. So this ovary got embedded, that got embedded in the thalamus. The ovary got embedded in the thalamus. And what becomes the edible part here? The thalamus, because all the food material got stored in the thalamus, not the ovary, right? This is the ovary which I have drawn here. Okay. So we are having this pericarp. Here you can see the mesocarp, and this is the endocarp. This is the endocarp, right? Now you were talking about the strawberry. This is the thalamus of the strawberry, this part. And these are the seeds. So again, all the ovules, all the ovary got embedded here. And part, the seed part is at this particular position, at the superficial position, right? The ovary get ruptured there. The ovary is very, you can say, a very small scale like things is left behind, but the seeds are present at the surface of the thalamus. This is the achene only, that all the ovary, this is the thalamus, and all the ovary like this, they are present like this. Okay, aggregates of the female reproductive part, that is the achene. Okay, so this is the false fruit example, strawberry and apple. Although in most of the species fruits are the result of fertilization, there are few species in which fruits develop without fertilization. Fruits are developed without fertilization. So what is that? Fruits are developed without fertilization. So that is called as, strawberry is a multicarpellary. Yes. Such fruits are called as parthenocarpic fruit. Parthenocarpic fruit. The fruit in which the fruit is formed without any fertilization, that is called as parthenocarpic fruits. Banana is one such example. Banana is an example of parthenocarpic fruit. Parthenocarpy can be induced through the application of the growth hormones. Okay, and such fruits are seedless. Seeds offer several advantages to the angiosperm. They are very, very advantageous to the angiospermic plant. Firstly, since the reproductive process such as pollination and fertilization are independent of water, seed formation is more dependable, right? Also, seeds have the better adaptive strategies for the dispersal to the new habitats and help the species to colonize in the other areas. As they have sufficient food reserve, young seedlings are nourished until they are capable of photosynthesis on their own. So young seedlings, they are having the seed leaves, that is the cotyledon. So they help in the process of establishment of the baby plant because they are having the capability to do the process of photosynthesis. The hard seed coat provide protection to the young embryo. Being product of the sexual reproduction, they generate new genetic combination leading to the variation. So these are the benefits of these seeds. They are having the genetic combinations after the sexual reproduction. Seeds are basis for our agriculture also. Dehydration and dormancy of the mature seeds are crucial for the storage of seeds, which can be used as a food throughout the year and also to raise the crops in the next season. So can you imagine agriculture in the absence of seeds or in the presence of seed, which germinates straight away soon after the formation and cannot be stored? So how long do the seed remain alive after they are dispersed? This was the question. No, agriculture would be impossible without seeds or with the seed that germinate immediately. And agriculture depends upon the ability to store seeds and the plant them at the chosen time, allowing for the plant crop cycles and the food security. So immediate germination would prevent storage, scheduling the planting season and the reliable food production also. How long do the seeds remain alive after they are dispersed? So this is a question, that how long they can be remain alive. Hmm? So they provide again, this period again varies greatly. The period is varies. We are having some examples. In few species, the seed lose viability within a few months. But in some large number of species, they can live for several years. Some seeds can remain alive for hundred of years. They can remain alive for hundred of years. This sentence is very important. There are several records for very old yet viable seeds. So what are the records? The oldest is the lupine, that is the Lupinus arcticus, excavated from the Arctic tundra. And the seed germinated and followed after the estimated record of 10,000 years of dormancy. You can see another record, 2,000-year-old viable seed from the date palm, that is the Phoenix dactylifera, discovered during the archaeological excavation at the King Herod's palace near the Dead Sea. So the seeds were viable after so long time. This Lupinus architects was viable for having the dormancy after the 10,000 years, and this Phoenix dactylifera 2,000 years. Hmm? So after completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions. So here are some questions. How many eggs are present in the embryo sac? How many embryo sacs are present in ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? And how many flowers are present on a tree, and so on? Can you think some plants in which the fruits contain very large number of seeds? So we are having answer of this. Let us see that, because these are very common questions. So 8 nuclei and 7 cells are present in one embryo sac. Usually one embryo sac per ovule. This is the usual condition. Varies greatly, one too many. One in the case of mango and 100 in the case of papaya. Usually one ovary per flower, but it can be compound or the multiple carpels. We have studied about the multiple carpels. In the case of somniferum, Papaver somniferum, varies enormously. What is that? Small tree, 100 flowers, hundreds of flowers on a small tree. And large tree, thousands of flowers were there. Plant with the numerous seeds per fruit. Papaya, you can see the number of seeds, 100 to 500. Watermelon, 200 to 800. Pomegranate, 600 to 800. Orchids, millions in tiny seeds. And dragon fruit, 1200 plus seeds in a fruit. Orchid fruits are one such category, and each fruit contains thousands of tiny seeds. This is the tiny seeds, and thousands in number are present in the orchid fruits. And similar in the case of fruits of some parasitic species like Orbanki and Striga. These are the examples of the parasitic plant, parasitic species, right? So how you seen a tiny seeds of ficus? Ficus is very, very important. How large is the tree of ficus developed for that tiny seeds? How many billions of seeds does each ficus tree produce? And can you imagine any other example in which tiny structure can produce such a large biomass over the years? Ficus is an angiospermic plant. It is not a gymnosperm. Ficus, in Hindi we call it anjeer or gular. Ficus seeds are indeed a tiny, about 1 to 2 millimeter in size. This is the answer of those questions. And ficus tree grow in a massive, reaching to height of 30 to 50 meters, 98 to 165 feet, widespread canopies. Their canopies are very, very large. A mature ficus trees can produce millions to billions of seeds annually. That could be 100,000 per fruit. And bamboo is another example that contain the tiny seeds, grow into massive grooves, producing enormous biomass. So now we will summarize the chapter. A seed forms after the fertilization when the ovule develops a protective layers. Zygote develops into the embryo while endosperm provide nutrition. Integuments of the ovule become the seed coat. A mature seed contains an embryo, stored food material, and a protective seed coat. The seeds remain dormant until the favorable condition trigger the germination. And the entire process of seed formation is called the seed development, which protect and nurture the next generation of the plants. So this is all for today's session. Thank you very much.